Hi everyone, on Saba and virtually around the world. Here we are again with another great evening of See and Learn. We're at a different location as we do normally. Tonight we are at the Busy Bee Bakery, which is doing a special venue for us. They're not normally open at night, but they're open to host us. And we're located in Windward Side in the main village of Saba. Tonight we're going to have a little mini presentation delivered by California Academy of Sciences, telling all of us about their Islands 2030 project and what it means to the region and to the world. After that, we have Dr. Susan Perkins, our expert on Anolis. She has been studying them on Saba for more than 25 years. So sit tight. We're going to be a few more minutes, but it's going to be a great evening. Thank you for watching, and thanks to Carib Trans for sponsoring our live streaming.
Hello, good evening, everyone, and welcome to See and Learn 2022. So tonight, we have a mini presentation for you guys to start off. We have Eudoxia from St. Martin's Nature Foundation, and we also have Lindsay from California Academy of Sciences, and they're collaborating together to support schools with the Environmental Learning Project. So we're just gonna have them come up for a mini presentation before our main presenter. Come up, ladies. You're welcome.
Do you want? Oh yeah. Oh, it's the clicker. The clicker. We do the presentation. Oh, right here. Yep. Sorry. No, this. I think it went to sleep. Oh. Hey, Susan, can you unlock your computer?
Stand behind of you.
I know there's lots of cords to trip on. Lots of cords.
Yes.
Kind of underwent a big strategic shift. We were looking at how we can engage our community members more so with science, because we believe nature is for everyone. And so just as a kind of a brief overview, we have programs and we work with every single elementary school in San Francisco, which is about 100 elementary schools. And they all come on field trips from kindergarten to 5th grade. And when we were approaching this, we thought to ourselves, how can we ensure that whatever it is that we build is relevant and meaningful to the people that are coming to us on our field trips? And so to do that, we did a lot of community listening, and we went and intentionally tried to find people who don't typically come to science museums or do science. Our science museum is a living natural history museum in the sense that we have scientists such as Dr. Lauren Esposito, who will be doing a night hike later this week, who do science and biodiversity research and evolution research all around the world. And the museum itself was started back in the year 1853 at a point in time where making natural history collections was an essential way of understanding the world around us. And so we have collections that date back over time. If you're a scientist, you might know this. Scientists go out into the field, they do a lot of research, they publish papers so that papers can get out into the world around us and to different community members. Lauren came to me about a year ago and said, okay, how can we do what you're doing in San Francisco, but with the communities where we're doing research? And so with that kind of question, we sat down and we came up with this project that I'm about to explain to you. Because since we believe nature is for everyone, and this idea of we have all of these incredible research collections that help inform conservation decisions, help inform all sorts of different things, how do we make sure that the people who need that information actually get access to it? Oh, I kind of already said that. Great. So we redesigned our strategic plan for the museum to think about biodiversity science, environmental learning, and collaborative engagement. And so what we're going to talk to you tonight about is the environmental learning research that we've been doing on the island of St. Martin. Our researchers have been here for a while in the Caribbean doing different things. And in 2021, something like that, our researchers connected with the Nature Foundation in St. Martin and discovered that they had an interest in building out environmental learning programs for the island itself. And so we came into this with this question of how might biodiversity science and education support St. Martin's schools, economy, and the environment around us. And then Eudoxia will talk about this a little bit more, but we went out and we did over 20 listening sessions with teachers, community members, and students to really understand what's motivating them, what are their questions, and how do we help support them. Yeah? What? Okay, your turn.
Yes, oh, that's my turn.
That's your
Okay.
turn.
All right.
Okay, do you want me to?
Next slide? Yes,
yeah,
you
okay.
click for me? Oh.
Wait, you don't need to.
So I don't think I need two mics. Good evening, everyone. I'm Eudoxia Williams. I was already introduced, and I'm the Education Project Coordinator from Nature Foundation St. Martin. I need to go back one slide. Yes. No. Yes. Yes, so as a foundation that is with a mission to enhance and preserve the local and natural environment for the generation now and for the generation to come. We've always had education or saw education as a key pillar to preserving our environment. Because why? Why nature education? Because if you look at the pictures, these are actual pictures of St. Martin. We put in a lot of efforts into a lot of work that we do. Oftentimes, we go out and we do beach cleanups, and you can see how much trash is collected. So we have like a successful beach cleanup. But can we truly speak of success if every month when we post on social media that we do a beach cleanup and we see so many trash collected, then something is wrong because, yeah, it's not getting any better. Also, on a wider, from a wider perspective, there is a lot of trees cutting because in St. Martin, there's a lot of building happening, and that leads to whenever there's like a lot of rain that comes down, we have flooding, we have landslides. So we are really observing that how, because persons may not be educated or due to ignorance, they throw certain things away, or they're not really conscious of what is happening around them and how their behavior impacts the environment. We all everyone understands that about climate change, but people don't understand that there is a direct link to the negative human impact on the environment. So that is why we thought we've always put a lot of attention to education. We do that through going into the schools, talking to the teachers, giving presentations, but we're a small organization and we have also limited resources. So that's why we're happy with the California Academy of Sciences, because this partnership with the Academy really helps us to have a more in-depth and stronger connection with the schools. And the idea behind of this, and I think we can go to the next slide, is that we want to create something. What that something is for the school, we didn't know at the start of the venture, but we wanted to create something that would really promote environmental learning and hopefully lead to a more better connection with the environment. So we wanted to support the teaching and learning processes, and we expect that after this, it's just a seed that we're planting. We're hoping to see if we can continue with this, but this is a pilot project. But we're hoping that with these efforts, it will lead to some type of adjusted human behavior that can bring changes to the environment. How we intended to do that? Through gap analysis. So we started with that in May 2022. We hit the ground running. Lindsay already mentioned it. We had different listening sessions with the schools. We spoke to students. We spoke to teachers, administrators. We also spoke to members of the community, just to get an idea of where do we stand when it comes to nature education. After that, the idea was that we would have ideas on things that we can develop for the schools in support of nature education. And we brought those ideas forward to the schools, to the teachers to get some type of feedback if that is something that they feel can work. And with their feedback, we went and we started to work. So in the month of July, August, September, we were busy designing, thinking out of working out the ideas that we had. And right now, actually this week, we're in St. Martin. We're going to try out the different activities. What those activities are, I'll explain a little bit later. And the idea is that once we try out these activities, we will get feedback from the schools and from the students, and with that feedback, we will evaluate the whole process. So what I'm going to talk to you about now is, we have the participating high schools. We have 8 high schools on St. Martin, and we have 5 high schools that participated in the listening sessions. Next slide. And like I mentioned before, we have other participants as well. So we spoke to student bodies. We had the St. Martin Youth Brigade. We also had afternoon school programs that we spoke to. We also spoke to some elementary schools. We spoke to parliamentarian, the Minister of Education. So we really tried to get a wide scope of every one. Next slide. So we did our analysis through listening sessions, and we specifically called them listening sessions and not interviews because the questions that we were asking, we were trying to ask questions that would allow persons to express themselves and not to guide them in a certain direction. So we were really looking to ask questions that would evoke stories and get them to just talk. So these are just a couple of examples of questions that were posed. What is something you really value about your community? what would you hope for kids to learn at school. That was specifically for the community members. When it comes to the schools, the teachers and administrators, we just wanted to know, what does a day of teaching look like? How do you get your students excited about science and environmental learning? And the students, we also asked them from their perspective, what science and nature education looks like, and how much do they learn about their local environment. Next slide. So I will explain a bit about what came out of these listening sessions. This is an overview of all of the topics that came up during the talks we had with the teachers and the students and the community members. So this is the overview of that, and as you can see, field trips are external visits. That was the highest scoring one. That means that from all the topics that we discussed, that was the topic that came up most of the time. And I will elaborate a little bit more on that right now. Next slide. So at 16.3%, what we learned is that learning about the environment, about your outside space, ironically speaking, that is mostly done out of the book. So both students and teachers indicated that they would not often go outside of the classroom, although from both perspectives they indicated that they would like to do it. The reason why they indicated that they're not able to do it is to make, in a nutshell, it would be time constraints. Teachers felt that because they have a curriculum to complete, they just have to stick to that, and there are not many possibilities to really go out there and explore, because it takes a lot of planning to do so. I'm thinking it was one more thing about that. No, it's back. Let me just check to see. Oh, yes, I got it. Another reason is that the teachers indicated that they would not go outdoors with the students is that they don't know where to take the students to. We don't really have a zoo in St. Martin. We don't have that many parks, so teachers really don't know where to take the students, where they can take the students to, or what they can do. Another thing with going outside is that we also notice that there is a difference. There are students who know a lot about the environment and who can really appreciate the environment, but those are students that from home, they are accustomed to go out and hike with their parents, or they would learn things about nature, especially about plants and the use of plants, as the bush tea, if you got a fever. So that's how some students indicated that they would learn about their natural environment. Next slide. Projects, hands-on learning, or inquiry-based or research-based learning. We just put that in one shot. I already mentioned it. Teaching is mostly done out of the book. And teachers indicated that they are a bit frustrated because oftentimes, like how in St. Martin you have different systems, school systems. So you have the Caribbean-based curriculum, the CXC, but you also have the Dutch system. And because they use these generic books, they would learn, for example, a school that follows the Dutch system, they would learn about a flower, but they would learn more about the tulip in the Netherlands and not about the flamboyant flower. So that makes a difference because they don't feel connected to what they are learning. So there is a need to have teachers do other things that is outside of the book, because the books that they are using are not locally based. Next slide. This was a shared second spot, at 15% connection with other organizations and schools. Teachers would like to collaborate more with other organizations in the community and have collaborative partnerships. They're very much interested in having the Nature Foundation come in and keep presentations because it just gives a different dimension to when they're teaching and when students are learning. And also teachers are curious about what others teachers are doing. It's a small island, well, it's bigger than Saba, but it's a small island. We don't have so many schools, but yet the teachers on the island, they don't really collaborate with each other. It's like all islands separate from each other. Next. Then we have the local curriculum or local expertise. And I already touched on that. Because they're learning oftentimes out of the book, they oftentimes don't have the materials about the local environment and what is applicable in St. Martin. So they're learning about the other parts of the world, but they're not learning about St. Martin. They've indicated that they would need they would like to see someone come in, like a local expert come in and teach their students about specific subjects. Also because in St. Martin, and I think that's the case a bit here in Saba too, from the conversations that we had here, in St. Martin, you have local teachers, but you also have teachers that come from abroad. So although they can teach about the environment, they don't know about what is really the St. Martin thing. So from the students' perspective, this is a quote from the Youth Parliament. Most English high schools follow the CXC program. Although the curriculum does have a Caribbean focus, you learn more about the bigger Caribbean countries, like Trinidad and Jamaica. There should be a subject that is implemented specifically to learn about the local history and current situation on the island. And I already gave the example with biology. When you learn about plants, you can learn about the tulip, but you don't learn about the flamboyant flower. Next slide. Lack of resources, teachers from and lack of new ideas. So they're trying, but they just don't feel that they have enough resources that they can tap into. It's a struggle to find local resources or materials about, let's say, a specific the specific lizards or anole from St. Martin. There is no information about that. So even if they're willing to go out there and try to make something, because we don't have much documented, they're basically, they feel stuck. Preservation and protection. It has been said that there is a disconnect between nature or the environment and what the people living in St. Martin seem to value. And therefore, it has been recommended that we should have a stronger curriculum for STEM-based learning, so that students from young can understand what it's all about, that it has more attention. There should be more incentives as well from the local government for STEM-based higher education and jobs. Oftentimes, when we look at the organizations that are busy with or work with the environment, they are oftentimes not locals that are working there. I put in a quote, we have to preserve nature. We are country people that are living in concrete cities now. So that was a sentiment of one of the community members. She felt that, St. Martin is oftentimes wants to be progressive in the sense of, yeah, we want to build, we want to attract, international persons, but we forget about the environment and how we do have to preserve that, so that there is a disbalance in that. Habitat destruction is the primary threat to terrestrial wildlife and plants on St. Martin, but that isn't something that is seen in a lot of environmental education. So no one really speaks about it. And I already mentioned it, the thing that is lacking is that people are not seeing the value of their natural environment. Next slide. Teacher training and development. So I already indicated that teachers spoke about the struggles that they had, about the fact that they're teaching out of the book, and they themselves feel a bit uncomfortable about that. They also want to go outside and have more hands-on learning, but they don't know how to. So there is a call for quality continued education. Teachers need to experience themselves how hands-on learning works, and with workshops that are really engaging and that will really empower them to put in that effort. From students' perspective, they also indicate that they should learn to make their lessons more engaging and hands-on. Many students indicate that there's a lot of focus on theory but little to no practical classes. Next slide. Loss of local history, heritage, and cultural traditions. I mentioned before that there seems to be a disconnect between man and nature. Many persons living on the island do not seem to value the country's local environment. People see and observe the changes, but there's nothing really done about it. And there was a quote that made a pretty good impression on us, is that it was stated that there is a great value in listening to children. Students do not think their lives are worth telling the story. It is important to give students information about their families and backgrounds and to show importance. Social learning is needed. It's not about bringing St. Martin to the future, but also rediscovering roots and culture. Next slide. And the last topic was more focus on nature education from a young age. It was recommended to start from primary or elementary school level, or even preschool level. But we did, from the different talks that we had with also teachers that teach in elementary schools, they actually do a lot, but it was funny to see that once the students reach high school, although they know what is right and what is wrong, they seem to forget about the norms and their values. So especially the reinforcement in high schools is a must. So with the designing and piloting of activities, what we decided to do is we planned some field trips activities for students from the various high schools. And with that, we will do it at the Emilia Wilson Park, for those who know St. Martin. It's a park that is pretty much accessible to all schools. It's close to the schools. And we are going to try different activities that they can do. And we would like to get then feedback from the students to see how they experience that. And at the same time, we're also keep doing a teacher training, a workshop, and we will be offering tools and best practices to empower the educators to actually go outside of the classroom and explore the environment with the students. The way ahead, we had other ideas as well that we can develop for the schools. For example, creating a map of St. Martin, showing suitable places where teachers can bring their students to. We're looking into creating a resource center. We're seeking funding for that, or we have just received funding for that, whereby, because that was one of the things that was mentioned, they have no resources, they don't have microscopes, so our idea is to allow teachers to come in and they can lend microscopes so that they can do their experiments, and we will then offer training alongside with that so that they know how to give those classes. And that's a bigger picture. We're looking into creating education units within the foundation to be better equipped to support the schools. Right now we have 2 core staff members, and the rest are project staff members. I'm, for example, a project staff member, so we're actually looking for a more sustainable means of income so that we're able to so that we have the manpower to go out there into the schools. That's it. Yes, thank you.
Okay, thank you to Eudoxia from St. Martin and Lindsay from California. Let's give them another round of applause, please. And now we will be continuing with our introduction for our main speaker tonight, Susan Perkins. So a big thank you to Carib Trans. It's one of our main sponsors, and they do our live stream, so a round of applause for Carib Trans. Oh, I don't have my clicker. The clicker? Okay. And the Flamboyant Cottage is our amazing sponsor for tonight. It is a beautiful 2-bedroom, 2-bathroom home with a pool and a view of 4 islands. Also a part of the raffle. And a big thank you to our Prince Bernhard Culture Fund and public entity Saba, because they're one of our biggest sponsors, so a round of applause for them. Thank you. And we have over 51 sponsors this year, so we have a lot of support and a lot of love. Thank you to all of our sponsors. A round of applause for them. So there's many ways to support us. You can buy a raffle, or for free, you can just write a review on greatnonprofits.org. It really means a lot. And again, raffles, raffles, raffles. These are the many, many prizes that you can win this year. Tennis lessons, beautiful bracelets, indigo dyeing. And these are our upcoming events at a glance. So on Wednesday, we have another presentation. We have with Susan Perkins and also Erin Mueller. On Thursday, on the Fort Bay, we have a field project. And Thursday, we also have another hike, a night hike with Lauren Esposito. So look out for these events. Don't stay home. Get active. And for tonight's speaker, do you know that anoles you see on the island are only found on Saba? But we find them almost everywhere, right? It is estimated that this little island has probably more than 70,000 anole lizards running around on our walls, in the forest, and even in your house. I'm sure some of you guys have seen them. Well, have no fear. They're your free pest control, right? They eat all the insects and the bugs in the home, so they help us out a little bit. But they also carry some baggage with them. Dr. Susan Perkins is here to explain the variety of the baggage our anoles carry and how her work with anoles is contributing to science. Susan Perkins is presently the Martin and Michelle Cohen Dean of Science at the City College of New York. She is a parasitologist who has been working in the Caribbean for almost 30 years, studying the malaria parasites of the lizards that inhabit the islands. An animal lover from New York, her field work has taken her across the Caribbean, but also in Africa. Pretty cool. Australia and Indonesia. She's been around. Susan was previously the curator of the microbiology at the American Museum of Natural History. She is the author of more than 80 scientific publications, as well as the book, Welcome to the Microbiome. So, act together And let's learn about our lizards by giving a warm welcome to Dr. Susan Perkins.
I steal just the clicker. Thank you so much, Alexis. That's the nicest way anyone's ever told me that I got around. Thank you so much, and thanks Eudoxia and Lindsay for setting that up. And I just wanted to share before I get into my work, St. Martin and New York City are not that different. I mean, we struggle with the same things in New York City. How do we connect our students with the natural world? So it's awesome what you're doing in St. Martin. I'm really happy to see Cal Academy participating. And I'm so thrilled to be here. As you learned, I have been doing this for 26 years. It seems hard to believe, and I think I've accurately counted now. I think this is my 17th trip to Saba over the years. I've been coming for quite a long time, and I'm going to not tell you all of that tonight, but give you a few highlights. And I wanted to share this, and this is partly for Tom and Helene. So it all started with my PhD advisor, Joe Schall, and his wonderful grad student, Chris Statz, who was practically a native Saban by the time she finished her master's degree. So Joe was a professor at University of Vermont, Chris was a master's student, and I had the amazing fortune to follow in Chris's footsteps and pick up, and you're going to hear about that work. So what I want to do is put pieces of this puzzle together. So I'm going to start by introducing you to the hosts. I'm a parasitologist. I have hosts and I have parasites. I'm going to start with the lizards and tell you a little bit about your amazing anoles, or anoles. So anoles are found all over the Caribbean basin. So all throughout the Antilles, Greater Antilles. We see them around South America, Northern South America, Central America, and of course we have some in the US as well. And some of them have been amazing invaders. Some of these anoles have traveled all the way to Hawaii, for example. How do we characterize what an anole is, or an anole? This male is helping us out with this big throat fan. The fancy name for that is his dewlap. And the males use that to do push-ups. They want to advertise that they are healthy and that they are ready to defend the territory, and they have these very colorful dewlaps. So as you go around Saba, I want you to pay attention, because Saban anoles are not uniform. You will see yellow, almost orange, and almost white in sabanus dewlaps, depending on where in the island you find them. Another thing that makes an anole is their feet. So they're not like geckos. They don't have super sticky feet, but they do have these toe pads that do let them climb up surfaces, and they're very good at that. They are different colors, and they sometimes can change colors. This is the same species. They often do this based on the temperature that they are. So they're not like chameleons in that they're camouflaging themselves, but based on how warm they are, their skin will expand. And all anoles eat insects. So as you heard from Alexis, this is your local pest control. They are chomping up insects, except sometimes when those insects are super large. And this was a photo I took on Dominica, and I was laughing when Lindsay showed the collection. I was like, oh, that's very similar. There's a Hercules beetle from Dominica. They know how big their mouths are. They won't push it. So an interesting thing was when I started my PhD, everyone thought that the anoles on Saba must have come from St. Martin, right? Because everything else comes from St. Martin, right? The groceries come from St. Martin. Our passengers come from St. Martin. Of course, the lizards came from St. Martin, too. And while I was doing my PhD, another researcher using DNA, using genetic data, actually showed that no, the anoles on Saba are more closely related to the anoles from Guadeloupe. And when you look at these guys, it makes a lot of sense. In fact, this anole here from Guadeloupe, we would call the giraffe anole. So we think this is the ancestor of the Saban anole, and you're going to see this a couple times, so lock that in your head. One of the projects that my grad student Brian did at one point was not on parasites. So before we get to the parasites, I want to just share some results of what Brian was doing, because I had already been coming here for more than 10 years at that point. And like I said, they have different dewlap colors. If you go to Sulphur Mine, the Anolis are a little bit bigger. They're a little bit tanner. Up on the Crispine, they're a little bit smaller. They're a little bit darker. They seem to have slight differences depending on where in the island they were from. So Brian actually used a form of genetic data called a microsatellite to look at populations. And I don't want to bog you down in too much deep science, but basically in our DNA, we occasionally have these repetitive regions. So here you see TA, 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 TA. Because it's repetitive, it slips when the DNA is copied as the cells divide. And that means that individuals have different numbers of these copies. So if we analyze these copies, the number of repeats, we can individualize somebody if we had enough. So early paternity tests, in fact, still some paternity tests, the O.J. Simpson trial, forensic techniques use this type of data to look at individuals and populations. And so Brian took the same approach. Wait till the motorcycle gang goes by. Okay. Motorcycle gangs are way smaller in Saba than New York City, so that was quick. Awesome. And I just want to show you what the expectation might be. So each of these boxes would be a site on the island. So we might have Troy Hill Road, Wells Bay Road, Crispine Trail, Booby Hill, etc. So if the genetics are Go back. There we go. If genetics are uniform, when we apply this method of microsatellites, we couldn't tell which site a lizard was from just based on the genetics that we see. Versus if there was specific structure, in other words, if the Troy Hill lizard stayed on Troy Hill, mated with other Troy Hill lizards, raised Troy Hill lizard families, etc., and they never went off exploring another part of the island, we would eventually see breakdown in terms of sites. And just in a quick recap, when we analyze these data from Saba and Anolis, there is complete what we would call panmixia. So the Anolis, even though on the short-term scale may look a little different, this large population of lizards is moving around, not as individuals probably, but over time moving around. There's a lot of what we would say is gene flow. They're interbreeding, etc. So we have a uniform population. But what was really surprising to us is that in Brian's study, we sampled 379 lizards, and just in their mitochondrial DNA, the little bit of DNA that is in our mitochondrial organelle we inherit from our mother, in just one gene, too, there were 113 different genotypes. That's an exceptional amount of genetic variation, which means, as Lynn asked me this morning and as Alexis told you tonight, this is a very large, robust population of Anolis. There's so many of them that they've accumulated these genetic differences. So the take-home message with the lizards is they move around, they may have short-term adaptation to their site, but they've been around a long time and there's a lot of variation. So, as I mentioned before, we are pretty sure now that Anolis sabanus colonized from Guadeloupe. And when these results first came out, it was a little puzzling to me, because remember, it seemed like St. Martin was the better, more obvious choice. But if you look back at the geological history of the Lesser Antilles, it makes a little bit more sense. And I think this is where some of our divers might have a better appreciation than us terrestrial folks. And that is that Saba, the part that's above the water right now, actually, when the sea levels were much lower, was a much bigger island, which we now can see reflected in the Saba Bank. So it's not unheard of then that Anolis from Guadeloupe would have drifted maybe after a hurricane and hit this larger island, the precursor of Saba today, and had a very large population that eventually retreated to the landmass that's currently above water today. And that probably happened about 100,000 years ago. So now let's switch gears for a little bit, and I'm going to talk to you about my work on malaria parasites. So I think most people know malaria, you learned this in school in your textbook, that malaria is transmitted from human to human by the bite of a mosquito. So a female mosquito feeds on blood. She needs the fat and the proteins to lay her eggs. If that person has malaria, she picks it up. The parasite develops in the mosquito, and then if she takes another blood meal, she will inject the malaria parasites back into the next person she feeds and start the infection in them. Okay? Malaria in humans is still a major global concern. So we have about 4 or 5 species that cause the disease in humans, but one of them is responsible for the vast majority of deaths. And there's still over 200 million cases every year. And although the numbers are falling, we have 500,000 people dying worldwide of malaria. We're making progress, I have to say. There are some vaccines, we have bed nets, we are definitely lowering the incidence of malaria, and that's great news. But the pressure we're feeling from behind is that our available drugs to treat malaria are starting to fail us. The parasites are starting to evolve resistance to those drugs. And so if we are not complete with our eradication and controlling of a disease, we may find ourselves in a scary future where the drugs that we have at our disposal are not particularly effective. And I think all of us, having just gone through COVID, maybe are a little bit shaky here. You know, we can't necessarily trust that we're going to be able to eradicate this. So humans, of course, are the major host in terms of economics and sociology, etc., of malaria, but we're not the only hosts. In fact, for someone like me that works on wildlife malaria, we're not even very interesting hosts. As I said, we had 4 or 5 species, but there are at least 60, probably hundreds and hundreds of species of malaria that infect birds. We have many, many other species of malaria in mammals, including other primates like this monkey, rodents, bats, and ungulates, and over 100 now, this slide's a little old, found in squamates, which are lizards and snakes. And this has been an amazing system for someone like me to study because it means I have gotten to travel all over the world catching lizards and sampling their parasites. So this is a map. All of these red dots are places where, as of around the late 1990s, different species of malaria parasites in lizards have been found. And what maybe could jump out at you is that unlike human malaria, which is confined to sort of the tropics and a little bit of subtropics, we see lizard malaria as far north as Wyoming, or you can't quite see it, I covered it up, but parts of Australia and New Zealand. So it's very diverse and found all over the globe. So I've been doing this for a long time. I've been working on lizard malaria for almost 30 years. And I can predict some of the most common questions. And of course, one thing you might be worried about if you've never heard of this system before is, oh my God, I just came to an island that's full of malaria. No, don't worry. Humans cannot get lizard malaria. Most malaria parasites are very specific to their hosts. Yes, some of the human parasites have jumped over from non-human primates, but for the most part, this doesn't happen, and there's no documented evidence of anyone getting lizard malaria or bird malaria, so don't worry about that. The second question is, all right, we're not worried about getting malaria, but maybe the lizards don't even need to worry about it. So a lot of people ask, do the lizards get sick from malaria? Part of this answer is a tough one because, and you will see if you've come out on a field project with me, you can't catch dead lizards, right? So if the parasite really was awful and killing the host, we wouldn't see it. That lizard would die. We have 2 systems of data. One was collected in California, and when lizards had malaria in California, it did affect a lot of their, what we would say is life history. male lizards could not hold down a territory. And both types of lizards, these are fence lizards, not anoles, but very close relatives, are territorial. They want to stake out their territory. If a male fence lizard had malaria, it couldn't do that. The male fence lizards have these bright blue bellies. It affected the color of them, which probably affected their ability to attract females. We saw more broken tails, which means that's an anti-predator strategy, which means those lizards probably are getting attacked by predators more often. The females with malaria laid fewer eggs, and if the lizards were stressed, they would often die. So, and I'm going to pump this ahead a little bit. So Chris Statz, my lab mate, spent at least, 6 months living here on Saba, traveling all over the island and trying to repeat the California study. And she was so surprised because she found something very different. So in neither system do we see fevers, right? Lizards are so-called cold-blooded. They can't regulate their temperature, but cold-blooded animals can get what we call as behavioral fevers. They'll go somewhere warmer, heat their body up in the sun, and try to kill the pathogens. But there's no evidence that lizards that have parasites did that more often. The Saban Anolis who had malaria still had territories. There wasn't any effect on the color of their spots or their dewlaps. There wasn't any difference in the broken tails. The females laid just as many eggs, and they rarely died from the infection. So it seems that the parasites that the Anolis lizards have in the Caribbean have probably been with them for millions of years, and they have adapted to exist in harmony. They do get infected. The parasites, as you will see in a second, do cause some harm to their cells, but the individual lizards can withstand it and survive and reproduce. So the other question, and this is one I really wish I knew the answer, is we don't know what transmits malaria from lizard to lizard. So if I were to go out by the Calicat, just a little bit north, with that system, it's not even a mosquito because it's too dry out there. It's a grassland. But sand flies that live in that area transmit it. It's very unusual. In the Caribbean, we don't really know. It's probably some kind of mosquito, but it hasn't been well studied, and any attempts I've made to find the vectors have not been successful so far. So we actually don't know the exact insect that does that. And now I just want to put these two systems together. So what have we been doing to look at lizard malaria in the Antilles? So when Joe and Chris Statz were doing their work, they were sampling the same sites around Saba for season after season. And one of the things that jumped out to them is that in different parts of the island, you see a different prevalence of the parasite. So what this figure means is that if they were collecting lizards along Troy Hill Road, 64% of them had malaria parasites. Versus if you went down to Sulphur Mine, this is before they bulldozed that track, if you went down and walked the old path, only 10% of the lizards had malaria. And St. John's, if you collected by the school, very few lizards had malaria. As we've continued to study this, we've realized that these relationships are not stable at all. So here are some new data from 2004, 2009, and 2014, and all I want you to get from this graph is that it's not predictable, right? So Crispine Trail, which had a lot of parasites, in 2004, it dropped off. Troy Hill had very few in 2004 and increased. And the Latter Bay Trail, we saw a lot in 2004, a lot in 2014, but almost no parasites in 2009. So there are these really interesting microhabitat dynamics going on in terms of the parasites found around the island. So back in 1995, when Joe and Chris were doing their work, they were only thinking of two species. There was one species called Plasmodium floridense, which from its name, you could probably guess where else it's found. Yes, Florida, but all over the Caribbean. And then there was another species that was really unusual. It was found in red blood cells. This is a lizard red blood cell, and here's the parasite, this little purple blob. I'm not tall enough to reach, so our viewers on live stream, that lighter purple blob is our parasite. It was thought that that parasite could also infect white blood cells. So here's a white blood cell, and here's the parasite. And that was very unusual, but it was thought to be that the parasite moved into white blood cells after the infection got going. So one of the first things that I wanted to do was add genetic data to this system. So I came down, I was collecting blood smears to look under the microscope, but I was also taking samples to extract DNA, amplify genes from the parasite, sequence them, and then use sequences to build evolutionary or phylogenetic trees to give me information about history of the parasites. And so I wanted to focus on this system of red blood cell parasites and white blood cell parasites. So when we compared just how they looked, ignoring the fact that they were in different cells, we couldn't tell the difference. So this was about the same size, etc. When we applied genetic data, though, it really jumped out to us. The red blood cell parasites, and these are found all over the Lesser Antilles, were their own group, and the white blood cell parasites were their own group. So in fact, the parasites in St. Kitts or Dominica are more distantly related based on where they are. And I want to remind you now of what I said about the Anolis, right? So we think the Saba Anolis came from Guadeloupe. The parasites support that hypothesis. The red blood cell parasites on Saba are identical to the parasites in Guadeloupe. The white blood cell parasites on Saba are identical to the white blood cell parasites in Guadeloupe. So this is additional backup information that we think that that's where they came from. So by 2000, we had now split these into two different species. Azoraphyllum is in red blood cells, and then a new one called leucocytica for leuco-white, leucocytica in white blood cells. So I just wanted to quickly show you these maps. This was part of my PhD work, just so you could see, again, this relationship. So we see azoraphyllum red blood cell parasites all over the northern Antilles, and then we see other types. So each letter is a different type of red blood cell parasite. But the white ones are very different, right? And this, again, I wanted to just show this to underscore that even though we have parasites on St. Kitts, the ones we have on Saba are exactly like the ones on Guadeloupe, and in this case, Dominica. Okay, for the last couple slides of this talk, I'm going to turn it over to my PhD student, Sarah Pangburn, and she can tell you what we've been doing in the last just couple years, and in fact, what we're doing right now on Saba when we're not doing presentations and field projects. Lynn's got us so busy, but we are having a blast. So I'm going to pause for a second, turn it over to Saba, and then I'll just wrap up and say thank you.
Okay, thank you. So as Susan has told us all tonight already, we can learn a lot about the lizards and their parasites themselves by studying them here, right? So we know more about their evolutionary history in terms of the lizard and the parasite. However, what I'm going to talk about is my PhD work with Susan, and that encompasses how we can study these lizard and malaria parasites and the mechanisms they use and how that can actually tell us about Plasmodium parasites in general. So that can even relate back to the Plasmodium parasites that infect humans. Okay, so there's a process that the Plasmodium parasite undergoes called hemoglobin digestion. And this process is basically a fancy way of saying that the parasite eats hemoglobin. So that parasite lives within a red blood cell, and within the red blood cell is the hemoglobin protein. So for humans and other animals, the hemoglobin protein transports oxygen around our bodies so that can help us function and breathe. So once the parasite is within that red blood cell, it can then take that protein and actually eat it in order to break it down to make its own proteins, which it needs to survive. And a byproduct of that is that by chewing up that hemoglobin protein, it releases free heme. So heme is an iron source, which can be cytotoxic or toxic to the cells. So in order to avoid that, they turn that free heme into a brown crystal called hemozoin. So as I'll show you in a minute, that brown crystal is actually one way that we can identify the parasites when we're looking at them underneath the microscope. And so this is what that looks like here. So this is a human malaria parasite in a human red blood cell. And that crystal that the arrow is pointing to is the hemozoin. So that's a byproduct of the parasite digesting the host red blood cell hemoglobin. And so this is important because a lot of the drugs that we use to treat malaria actually involve this process. So one drug that you may have heard of is artemisinin, and that's currently our main forefront drug for treating human malaria. And it works by basically getting activated by this hemoglobin digestion process. So as the parasite digests hemoglobin and releases the free heme, that free heme actually activates the drug, artemisinin, which can then go on to kill the parasite. And so that's how we treat human malaria. However, if you might remember with what Susan said, there's currently drug resistance rising in these human malaria parasites. So it's important for us to study this process a little further to learn as much as we possibly can about it. So my question is, do lizard malaria parasites also eat hemoglobin in red blood cells in lizards? So the way that we're doing this is by looking at these Saba anoles that we have here. And this is our lab setup at Flamboyant Cottage. So within the lizards here, there are 3 main species of parasite, as Susan explained. So the first one is Plasmodium azurifilum, and this is a lizard red blood cell here. This is the lizard red blood cell nucleus, and this is the parasite. Now, Plasmodium leukocytica, as she told us, is within a white blood cell, so this is the lizard white blood cell, and this is the parasite here. And Plasmodium floridense, lastly, is also in a red blood cell, and this is the parasite over here. Okay, so for Plasmodium floridense, we actually do see this hemozoin. So that means that this parasite is digesting hemoglobin. And so I have it pointed out with the arrows, but once again, this is the parasite here, and this brown pigment is the hemozoin. However, if we look at Plasmodium azurifilum, which is also in a red blood cell, so it has access to hemoglobin, we don't see any brown pigment or any hemozoins that could indicate that that parasite is not doing this process. So this is the parasite here, and you'll notice that there's a lack of that brown crystal. And lastly, Plasmodium leukocytica is in a white blood cell. So this is the enlarged picture of it, and this is the parasite here. And there's no hemoglobin in white blood cells, so they're not digesting hemoglobin, and therefore there's no hemozoin. So if we go back to human malaria parasites, they use approximately 17 genes in order to complete this hemoglobin digestion process. So what I want to know is, do lizard malaria parasites have these genes for the same process? So do all 3 of these parasites that live in the Saba anoles, do they have the same genes that the human malaria parasites have? So Susan explained to us her use of DNA with the mitochondrial microsatellites, as well as her phylogenetic work. So that's looking at the genome of the lizard or parasite, whereas what I'm using is actually something called RNA, and I'm looking at the transcriptome. So this RNA basically tells us, of the entire genome, which genes are the parasites using and turning into RNA, because that RNA will be further turned into proteins, which will then be used by the cell. So with my work that I've been doing with these Saba anole samples, basically I've seen so far that there's evidence for 11 of these hemoglobin digestion genes in Plasmodium floridense, Plasmodium azurifilum, and Plasmodium leukocytica. So all 3 of these parasites have the genes that are involved in this process. So for my future directions, I want to know, how similar are these genes to the human malaria genes? And are these genes, will they be maintained in the genome of the 3 parasites, even though some of them, 2 of them, are not digesting hemoglobin? So will they still have them in the future? Or are they possibly being used for something else? So if you remember, I told you that hemoglobin, which is depicted by this, red 4 circles, that's being digested to be turned into proteins by the parasite. So could the parasite be using these same genes that are involved in hemoglobin digestion in malaria parasites to digest other proteins other than hemoglobin to make their own proteins? And with that, we would both like to say a huge thank you to many people.
So I'll come back to this. These are a whole bunch of people that have helped us with our work here on Saba and been analyzing. I'll let you click, Sarah. I really want to give a particular special thank you to Sherry Martz and Larry Holler at Flamboyant Cottage. I couldn't ask for a better lab. It is awesome. It comes with a pool. It comes with a bartender. It comes with witty dialogue at all hours of the night. And then if you click the next thing, it has even come with a field assistant. So Sherry and Larry not just host us, but Larry is an amazing field assistant. And then I wanted to put this slide back up. I've been coming here for 26 years. And one of the things I love about this island is that every time I come, there's something different. There's a new store, there's a new restaurant, there's something new and fun, but yet it's not completely different. It's been an amazing community. I'm looking at Tom and Elaine. My first permit to collect was a letter from Tom saying, Susan Perkins can collect Anolis lizards here and take them home. That was my first permit. It's just been home. As you saw, these parasites exist all over the Antilles. We could do this work in a lot of places, but I would never think of a better place to do it than Saba. And it's been my honor, this is my third See and Learn, to keep coming back, seeing some old faces, meeting new folks, meeting folks who are just here for See and Learn, and getting to share what we do here. So I'll close out and hand it back and say thank you so much. There you go, Alexis.
Thank you again, and give it up for Susan. Amazing presentation. And we just want to say thank you to everyone for coming. Thank you to Busy Bee for hosting us tonight. And give it a round of applause for yourselves. We also have field projects tomorrow. We have a hike tomorrow night. So please don't be afraid to come by the tent. We're there from 8 a.m. to 4 p.m., Monday through Friday. So come, sign up, support, and thank you again.
Yay!
Miami is known as an international community and the hub of trade and logistics for the Caribbean and Americas. A leader in the freight logistics industry, CaribTrans has served the Caribbean region for more than 30 years, with a footprint that has grown from one island to now serving the majority, as well as Central and South America. CaribTrans is an NVOCC, a non-vessel operating common carrier, meaning it has the same responsibilities as a shipper without owning the vessels or planes. Its primary customers, individuals who ship clothing, electronics, and other personal items, and occasionally cars.
Most of the stuff that they're sending is because they don't get it there. So for us to provide it on two services, air and ocean, we can give it a choice of how fast you want it. So if you want it really fast, we're going to send it by air, you're going to have it next day, sometimes even the same day.
That on-time service has been the key to CaribTrans' success, whether shipping by air or by sea. The company moves about 5,000 TEUs of freight each year, more than any NVOCC in the region. And since it joined the Saltchuk family of transportation companies a couple of years ago, CaribTrans can offer customers even more shipping options.
Other NVOCCs, they depend on other shipping lines that are not part of their family. So they need to rely on whether they're going to sell or not, whether they are late or not. It doesn't happen to us because it's part of our family of companies. We control the service that we give.
Sí, buenos días, por favor, con Diana.
Quality service is what keeps customers coming back. Customers are treated like family, and when they leave to research competitors, it doesn't usually last long.
They have left and they have come back because they've said that, nobody does it better than Carib Trans. So because of that relationship.
That exceptional service includes consolidation. Customers can have freight sent here and stored for free for up to a month, then consolidated and shipped on to the islands and Americas, a perk for the region's growing small business market. Puerto Rico is an expanding market for Carib Trans, about the size of Connecticut and rich in history and culture. Puerto Rico is home to many pharmaceutical manufacturers, as well as a thriving tourism industry for which Carib Trans moves raw materials and supplies. But serving small businesses is still the company's bread and butter here in San Juan. Businesses like Caldera Café, a local coffee company. Owner Alfredo Rodriguez started growing coffee beans at his mountain farm about 20 years ago. He recently ordered a variety of new equipment for his farm and shop. He uses Carib Trans because it consolidates shipments, which cuts down on the number of times he has to pay the Puerto Rico import tax.
Not many companies want to do that. They want only big cargoes and maybe half container or full containers, but they don't want to consolidate because it's more difficult.
Turning challenges into opportunities is what Carib Trans does, and that includes helping to grow small businesses like Caldera Café by taking the hassle out of shipping.
We can be a gateway to expand their business in the Caribbean and back to the U.S. And wherever they can sell their products, we are also going to be a partner for them.
That customer dedication has made Carib Trans what it is today. Now, as part of a larger family with more transportation resources, Carib Trans hopes to grow beyond its current market, offering services to additional Latin American ports and eventually Europe and Asia. But even when it becomes a global logistics company, Carib Trans wants to stay true to its roots, always providing personalized, reliable customer service and a commitment to the communities it serves.